Thank you. So we're here. The cut section of a human DNA is inserted into a cut plasmid and they are joined together to form a what? Which plasmid? Recombinant plasmid. Very good. Please be sure of the spelling. Recombinant plasmid or recombinant DNA. These plasmids are inserted into excuse me? They're inserted into what? Into another bacteria, yes. Okay. Remember that we use two bacteria. One, we get the plasmid from one, and then when we create the recombinant DNA, we put it in another bacteria. So it's going to be inserted into another bacteria, and replication occurs. The process is used to produce human insulin. Very good. This is the typical example that is mentioned in your syllabus that is used to treat people with diabetes. In addition to genetic engineering, enzymes are also useful in other biotechnologies. Give two examples of how enzymes are useful. We'll answer this question. In biological washing products, in food, what am I writing here? In products, okay? If I want to create lactose-free milk, in juice extraction, the pectinase enzyme, I highly advise you that you write the name of the enzyme. We use here lactase enzyme, and we use here the pectinase enzyme. And in biological washing powders, we can say we use the protease enzyme to break down large and soluble molecules to small and soluble that can be easily washed away. Okay, the fermenter, and we answered it? Good. Explain why there is a water jacket. I think we answered it. Optimum temperature to control the temperature so that it doesn't denature, so that the enzymes, they work at the maximum rate, maximum enzyme substrate complex, and all the story. Okay? This is the mass of the fungus and the yield of the antibiotic during the fermentation process. So you have here, always look at the axes. Time, and here the mass of the fungus, and here the antibiotic production. So this X is the fungus, XX is the growth of the fungus, and here the growth of the antibiotic. Good. State the time interval over which the fungus grew at its maximum rate. The maximum rate was here. Yes? From where to where? Fungus. Where did you get 40 to 50? This is the growth of the fungus. The penicillin is this. The fungus. State the time interval over which the fungus grew at its maximum rate. The time interval, I think it's from 120 hours to 160 hours. Yes? Rate, then this is the maximum rate. This part. This is the part where the fungus is growing quickly. This small part, yeah? Yes, 40, 50, this part. This is the maximum, like at the end. The rate is how fast it was going. So we will look at this part. Okay? And let's make sure. If we're confused, always let's make sure. Anyone does mistakes. It's very normal. We're right? We're right. Good. And then they gave us the other answer. State the type of nuclear division. This is not a bacteria. This is a fungus. It has a nucleus. So don't say binary fission. It's not a bacteria. It's mitosis. Okay? Nucleus. It has a nucleus. Bacteria has no nuclei. Good. Explain why the growth of the fungus slows down. Why does the population of the... Exactly. We have limiting factors. Please, they love this word. They have limiting factors. So this is one word. And give examples of limiting factors. Here, excellent. Anything of what you said. There is a decreased supply of nutrients. There is accumulation of waste products. It became too acidic. It became too hot. Overheating. There is decrease in the nutrients, decrease in the space, whatever. Okay, but mention the word limiting factors. Good? Let's finish this exam and then we can revise reproduction. State the evidence from figure 5.2 that shows that penicillin is not needed for this growth. Look here. The fungus, it stopped at a certain time. But even though the fungus stopped, the yield of the penicillin kept on increasing. Yes? Let's read the question. State the evidence that shows that the penicillin is not needed for this growth. Penicillin is not needed for the growth of the I don't know what. The penicillin is not needed for the growth. The fungus here is growing without the penicillin. Okay, so the what is the P thing? Shows the mass of the fungus and the yield of the penicillin during the fermentation. At the beginning, there was no penicillin, even though there was fungus. What is your defense? What are you saying? Which is what repeated? The fungus will grow even though there is no penicillin, because it's the fungus that is producing the penicillin, not the opposite. Do you understand? The fungus is producing the penicillin. So the fungus will grow even though there is no penicillin. There doesn't have to be penicillin for the fungus to grow. Clear? Let's write a decent answer from the mark scheme. The fungus will grow even when there is no penicillin produced during the first 20 hours. Now, these are the questions that can be confusing. If you're confused, read the question carefully. Don't do like me. I go directly to the question. Read the story from the very beginning. Okay? Because I just don't want to waste time. Okay. Use the information in figure 5.2 to suggest why they did not allow the fermentation to continue for longer. They started to die, okay? No more fungus is there. And even the penicillin, it became constant. So they don't keep the microorganism in the fermenter forever because it doesn't produce forever and it doesn't react forever. Good? Is this exam finished? Suggest the downstream processing refers to all the processes that occur to the contents of the fermenter after it is empty. This involves making penicillin in a form that can be used. Suggest why downstream processing is necessary. Suggest means critical thinking question. Why do I have to downstream the process? And read the question. Here it occurs to the contents, to the final product, I mean, after it is emptied. Yes. Why do I have to do some downstreaming? Give me an answer. I want 3 reasons. Read it. Downstream refers to all the processes that occur to the contents of the fermenter after it is finished. The process is finished, I'll get the contents. Here I have the fungus and the antibiotic and everything together. Of what? Yes, okay, I will purify it. I will clean it. I want the penicillin alone. I don't want penicillin and fungus. Yeah, so I'm going to separate. I want 3 reasons. I can separate the fungus and the penicillin. I will purify. I'm going to prepare it for selling. I'll put it in bottles. I'm going to separate the enzyme. I'm going to remove the waste products. You have any answer? What else? I will remove the waste products. I'm going to filter it. I only want the penicillin. I will separate the fungus from the penicillin because I'm going to put the penicillin in bottles and I'm going to sell it in the market. Okay? I'm going to extract the penicillin. Any one of those. Let's look at the answers to get more ideas, if there are more ideas. Purify, separate, remove the waste products, putting it into pills because I'll sell it, adding color to it, anything. Good? The suggest questions are critical thinking. Explain why antibiotics such as penicillin affect bacteria but not viruses. Now, this is a very old question. Exactly. No cell wall. I want another answer, 2 marks. Huh? They're non-living, no cell wall. The viruses, they don't do respiration. There's no metabolic activity in the virus except when it is inside a cell. Okay? So the virus, no cell wall, no protein synthesis. It does not reproduce except when it is inside a cell. It is non-living. It has no metabolic reactions. Good? Any questions? Easy? Let's solve this one. This is also easy. Now starch. What is the enzyme that will break down starch? Into? Yes, maltose, not glucose. Okay? Maltase will break down maltose into glucose. Fat into perfect. Protein. Protease into excellent. Amazing. Another graph. Let's start revision. Compare the effectiveness. Use the graph and break it down. Use the graph and break it down. The easiest way to break down a graph is to go to a certain number, like for example, temperature 40, and compare one graph and use that same temperature and compare the other graph. Clear? Nice? Okay. We take a break and revise, or we revise straight away? You hurt my feelings.